Bismillahirrahmanirrahim. Dear students, Assalamu alaikum. I am Makar Ahmed and this is the lecture number 3 on ribosomes. In the previous two lectures, we have studied about some basics of ribosomes. Today we will study about the biogenesis of ribosomes, that how the ribosomes are synthesized. So the ribosomes, they are not the self-replicating particles. Ribosomes are not self-replicating particles. So if the ribosomes are not self-replicating particles, so it means that the ribosomes, it will synthesize from some genes. They are synthesized from genes. The information come from genes. So as you know that the composition of ribosome is that the ribosome, it consists of the ribosomes consist of ribosomal RNA and proteins. So the ribosomal RNA and proteins, so both it come from DNA. The DNA, it contains gene sequences for ribosomal RNA and it will form messenger RNA, and then the messenger RNA will form proteins. So combinedly the proteins and ribosomal RNA, it form ribosomes. The biogenesis of ribosomes, it is different in prokaryotes and eukaryotes. The 70s and 80s biogenesis of ribosomes. Biogenesis is different. So today we will study about the biogenesis of 70s ribosomes. So the first is the biogenesis of 70s ribosomes. As the ribosomes in eukaryotes, we say that the ribosomes are synthesized in nucleolus. In prokaryotes, there is no nucleolus. So in prokaryotes, the ribosomes, they are synthesized in cytoplasm. In prokaryotes, the ribosomes are synthesized in cytoplasm. Synthesized in cytoplasm. In the prokaryotes, in the bacteria contain the chromosome in nucleate form. The nucleate, this is the chromosome of bacteria. So this nucleate, it is in free form in cytoplasm. The nucleate, it contains DNA, RNA and protein. And these proteins are the non-histone proteins. These proteins are the non-histone proteins. So the E. coli, it contains circular chromosome. The nucleate of the chromosome, it will be circular and single. Haploid chromosome. Haploid chromosome. And the number of genes in E. coli or in bacteria, this is also in hundreds. The number of genes in prokaryotes are in hundreds. The exact figure I don't remember, but it is in hundreds. So when the biogenesis of ribosome occur, so the biogenesis of ribosomes, it occur from the genes on chromosome. So only 0.4% genes on chromosome are devoted for ribosome synthesis. For ribosome synthesis. The prokaryotes, it contain also the other types of RNA, but 80% of the RNA, 80% of RNA in E. coli is ribosomal RNA. So it means that the content of ribosomal RNA, it is high than all. Now, how the synthesis of or how the biogenesis of this ribosome occur? The ribosome, which is ribosomal RNA, the ribosomal RNA, it is the more stable. It is more stable than other types of RNAs. The other types are, for example, the messenger RNA and transfer RNA in prokaryotes. So this one is the more stable form. In the transcription of ribosomal RNA from the genes from which the transcription of ribosomal RNA occur, so such transcription is called, it is transcribed in the form of perpetual transcription. In the form of perpetual transcription. Now what is perpetual transcription? Perpetual transcription mean that there will be no change in the manner of its transcription, not changeable, but in regular manner, it is synthesized. Now the genes of ribosomal RNA, they are present in the form of operon. They are present in cluster form. The genes are present in cluster form, which is called, that is called operon. Now what is operon? Operon is actually cluster of genes. Operon is cluster of genes. Or we can define that when many genes are transcribed, when many genes are transcribed from single promoter, so this is called operon. For example, in E. coli, if there are 3 genes, this is the gene 1, this gene 1, it is responsible for 5S subunit, this one is responsible for 23S subunit, and this one is responsible for 16S subunit. So, and there is only single promoter, this is one promoter. So when the RNA polymerase come and binds here, so it will transcribe all the 3 genes simultaneously. So such type of genes, such type of structure is called operon. So operon is the cluster of genes. So here also the cluster of genes, they are transcribed only as a single unit. For example, when they are transcribed, so there will be one long chain of ribosomal RNA. Now this long chain, it contains the 5S, it contains the 23S, it also contains the 16S. Now these 5S, 23S and 16S, they are premature form. They are in the premature form, as I have mentioned. So I will try it again. So they are in premature form. For example, this is 16S, this is the 23S, and this one is the 5S. Now this is the premature form. Now it will be matured. So how it is matured? So the tailoring process occur with it. Tailoring with it. Now what is tailoring? So what the tailor do? Tailoring mean that some cutting will be occur, the pressing will be occur, and the sieving will be occur. So what happen here in the tailoring? The tailoring mean that methylation and cutting of these RNAs occur. RNAs occur. So first of all, the 16S ribosomal RNA will be isolated. First, 16S ribosomal RNA, it is isolated. For example, this is the long subunit. So this 16 one, it will be separated from this chain. The 16S, when it is separated, so this 16S, it contain 100 extra bases. It contain up to 100 extra bases. So it will belong then the mature ribosomal RNA subunit, the 16S. So these 100 extra bases, it will be excised, or it will be removed. The extra bases will be removed, and also the methylation will be occur. The methyl group will be attached to it. So the methylation is occur, and it will become now a mature one. Now when the methylation occur, then the proteins will also combine with it. Some proteins will also combine with it, and now this 16S, it will become mature. So this is the first one, 16S, and also the other three subunits. For example, this one is the 23S. The 23S, it will also be separated, it will be methylated, and it will be matured. This one, 23S. And then also in the last, the 5S. So the 16S, the 5S, and 23S, these ribosomal subunits will be formed. When the ribosomal subunits are formed, then the proteins are also required, the other proteins. So the proteins, they are synthesized from DNA also as messenger RNA. So the messenger RNA, they also synthesized as polycystronic. Polycystronic mean messenger RNA formed in two forms, the monocystronic and polycystronic. Polycystronic mean that when more than one type of messenger RNA are present along, so this is called polycystronic. For example, there are total 54 proteins are needed for ribosomal RNA to form mature ribosomes. But all of these 54 ribosomes, they are formed as 11 chains of messenger RNA. So these polycystronic, the messenger RNA tend to be polycystronic. So and polycystronic mean that several proteins in a single messenger RNA molecule. Polycystronic mean that several proteins in a single messenger RNA. So this is polycystronic. So when the proteins, they are formed, the proteins will also, they will bind to the RNA subunits, and it form mature ribosomes. Now how the translation of proteins are controlled. So the translation of proteins, it is controlled by autogenous regulation of translation. So here is autogenous regulation of translation. So autogenous regulation of translation mean auto means self, genus means synthesis. Now some self proteins cause the inhibition of overexpression of proteins. For example, when the need of protein is fulfilled, then one of the protein, that is large protein 4, the L4 protein, it binds to messenger RNA to inhibit further translation. For example, this is a long chain of messenger RNA. So to this long chain of messenger RNA here, when the L4 binds, this is L4. So when the L4 binds, so there will be no translation of messenger RNA further. Now why the translation is inhibited? Actually the translation is inhibited when there is no need of protein synthesis for ribosomes. So in this way, the ribosomes, the final two subunits formed in the prokaryotes are the 30S and the other is 50S. The 30S and the 50S, when they bind, so combinedly it forms 70S in prokaryotes. So this is about the biogenesis of ribosomes in prokaryotes. Thanks. Stay with me for the next lecture, that will be the biogenesis of ribosomes in eukaryotes.